All right, today we're doing something a little different. We're not just going to learn about science. We're going to crack a case, a real-life medical mystery. We're stepping into the shoes of some of history's greatest scientific detectives to solve a crime that haunted humanity for, well, centuries.
You know, for thousands of years, people would get these recurring violent chills and fevers. We call it malaria now, but back then, they had no idea what was causing it. All they knew was that it was deadly. So think about it. This was the ultimate cold case. The culprit was completely invisible. How do you even start to solve that?
Well, when you're trying to solve a mystery this big, you can't just guess. You need a reliable set of tools. And in science, that toolkit is called the biological method. It's basically the step-by-step process scientists use to turn a giant question mark into a solid answer. It's the very same playbook we're going to follow to finally unmask our killer. Okay, so let's break down this playbook. It's got 6 key steps. First, pretty obvious, you have to recognize there's a problem in the first place. Then, you start gathering clues. You make observations. Based on what you see, you'll form a hypothesis, which is really just a fancy way of saying an educated guess. From that guess, you make a logical prediction, a deduction. And here's the most important part. You test it with an experiment. Finally, you look at your results, draw a conclusion, and tell everyone what you found. That's the method. Simple, right?
Now, when you're gathering those clues, those observations, they come in 2 main flavors. You've got your qualitative observations. These are descriptive. Things like the patient feels cold or the fever comes in waves. Then you have quantitative observations. This is all about the hard numbers, the measurable stuff, like recording a patient's temperature as exactly 104 degrees. To solve any case, a good detective absolutely needs both.
All right, the investigation begins. First things first, we have to answer the biggest question of all. What is actually causing malaria? Who or what is our prime suspect here? You know, the early clues were, well, they were really confusing. So observers noted the chills and fevers, for sure, and they saw a clear connection to marshy, swampy areas. In fact, the name malaria comes from Italian for bad air, because they thought it was some kind of poison gas coming off the swamps. But here's the kicker. They realized people who actually drink the marsh water didn't necessarily get sick, so the killer was in the marshes, but it wasn't in the water.
And then we get the first huge break in the case. It's 1878. A French doctor, Laveran, is looking at a patient's blood sample under a microscope, and he sees something that just doesn't belong there. This tiny, strange microorganism. He looks at another patient, and another, and there it is again. He calls it Plasmodium. At last, we have a suspect right at the crime scene.
And this leads us straight to our hypothesis. This is that single, testable statement at the very heart of the investigation. It's the moment the detectives point their finger and officially name their prime suspect. Plasmodium, they say, is the cause of malaria.
But here's the thing about good science and good detective work. Having a suspect isn't enough. Not even close. You need proof. You need evidence that's beyond a reasonable doubt. You need a rock-solid experiment that can prove the connection and rule out any other possibility.
And this is where the genius of a controlled experiment comes in. It's so elegant. Scientists took 2 groups of people. First, an experimental group of 100 patients who definitely had malaria. Then, a control group of 100 perfectly healthy people. The only significant difference between these 2 groups was the disease itself. Then, they took blood samples from every single person and put them under the microscope.
The result? It was absolutely undeniable. Every single one of the 100 malaria patients had Plasmodium swimming around in their blood, and not a single one of the 100 healthy people did. The evidence was just overwhelming. Case closed. Plasmodium was our killer.
But wait, there's a twist. Knowing who the killer is, that's huge, but it isn't the whole story. The next big question was just as important. How was this Plasmodium getting from one person to another? Our killer had an accomplice.
So in 1883, another physician, A.F.A. King, laid out this amazing list of 20 observations he'd made. Let's just look at a few. He noticed people who slept outside got sick way more often. People who slept near smoky fires, which, keeps bugs away, they stayed healthier. And here's the smoking gun. People who use mosquito nets were protected. All the clues were pointing to a flying accomplice.
With these clues piling up, the next hypothesis just fell right into place. It's like a detective figuring out the getaway driver. Scientists made their next big claim. Mosquitoes are the accomplices. They are the ones transmitting Plasmodium.
And the final proof? That came from a British doctor named Ronald Ross, who did these incredible experiments with sparrows. He let mosquitoes bite infected birds, and then he watched as the Plasmodium multiplied inside the mosquito and moved into its spit, basically. When those same mosquitoes then went and bit healthy sparrows, you guessed it, the birds got malaria. The entire criminal plot was finally mapped out for everyone to see.
So what happens when a scientific case is solved this thoroughly, when the evidence just becomes totally overwhelming? Well, in science, our understanding actually graduates to a whole new level of certainty.
A hypothesis that survives round after round of tough, rigorous testing and is backed up by mountains of evidence from all different directions, it becomes a theory. And this is so important. A scientific theory is not a guess or a hunch. It is one of the most reliable, well-supported explanations that we have for how the world works. And if that theory holds up against every single attempt to prove it wrong, for years and years across countless experiments, it can become a scientific law or principle. This is the highest level of certainty you can get. It's a constant, uniform fact of nature. The case is officially closed, and its findings become a fundamental part of the rulebook for all future science.
Now that biological method we just walked through, the one that solved the malaria mystery, it's timeless. It's still what scientists use every day. But while the method hasn't really changed, the tools our modern detectives use, oh yeah, they've been seriously supercharged for the 21st century.
You know, even back then, math was a surprisingly powerful tool. Ronald Ross noticed that in his bird experiments, about 6 out of every 10 sparrows that got bitten got infected. So using that simple ratio, he could predict that if he exposed 50 sparrows, probably about 30 of them would end up getting malaria. It sounds simple, but that use of proportion is a basic kind of data analysis.
Fast forward to today, and we've scaled that up in a way they couldn't even imagine, with a field called bioinformatics. Honestly, think of it like swapping out a magnifying glass for a supercomputer. We use these powerful algorithms to analyze enormous sets of data all at once, like the entire human genome. It's the same detective work. It's just that now we have tools powerful enough to solve mysteries those early scientists could only ever dream of.
And really, that's the big takeaway here. The exact same logical process that unmasked an ancient, invisible killer is what scientists are using right now to tackle our biggest challenges, from trying to cure genetic diseases to understanding the mysteries of the human brain. The method works, it endures. So the only real question left is, what great mystery will it solve next?